Hello, good morning. Hello again. Do you have a good rest? Okay, great. So, my name is Diogo again. So, now I'll bring you today some slides about understanding our biology body. So, yeah, thank you. So, do you still remember what it is a cell and where it is and how does it work from yesterday? So let's just, in one slide, make a quick summary of it. So imagine that you are again in the library and you go to the visual system section. So, and you see the bookcase of the eye. And you pick up a shelf and you see the retina layer of cells. And you see there are many books. Some of them are copy of each other, so they have the same information. And others are different, so they have different information. That's the book, so the cell. Oh, sorry. So the book, the cell, and then you open the book and you go to the content, the text, and you see that it has many chapters, so chromosomes. Okay, and the chromosomes, the chapters have many words, the genes, and the genes have many letters, the DNA, and the DNA has the message. Okay, and you can open the book and rewrite a sentence in a piece of paper and you transcribe it, okay? And then you translate it to another language and you give a meaning of the gene to a protein, okay? And then the protein will have a specific role in the cell or in other cells as the translated message as for people from other languages that can interpret this message. We talked a little bit about cells and the immune system as well yesterday, and today we will have a picture of how organs are integrated in the production of these cells and the immune system and the immunity itself. So our agenda is how does our body work, and we will go through the human body systems, in specific the cardiovascular and lymphatic systems, and then the hepatic function and the enterohepatic circulation, because the liver is very important in uveal melanoma, as you know. And the process of cancer and metastasization, okay, and we will go from the process very quickly, just to have an idea of our understanding of this process. And we will finish going through what do blood tests check, and if we get some summary of these three topics. So our human body have several systems. So from the skin, so the integumentary system that allows us to protect the body, to keep the temperature, to make a first barrier to external factors. The skeletal system that allow us to move and have a structure, okay, with the bones. The muscular system that allows us to move. The lymphatic system that we will go to see in further detail, the respiratory and the digestive system to breathe and to digest the food that we take. The nervous system that allow us to control our body, okay, with a brain and the nerves that move all the muscles in our body. The endocrine system that essentially is constituted by the organs that produce some proteins or other kind of messages that are released to bloodstream and go to all the body to control temperature, to control production of some essential elements to life. And the cardiovascular system that we also look in detail after these couple of slides. And then the urinary system and the female and male reproductive systems. So the cardiovascular system, it works like actually a roadmap. So this is our beautiful country, Portugal, and you have like the highways that where the cars and in this case the blood elements go faster, okay, and you have the big arteries and the big veins, and then you have the smaller arteries and the smaller veins where the speed is slower. And the circulatory system is composed mainly by the heart, the blood vessels, and their elements. So the blood, which is composed, as we will see, by two different groups of components. So if you just collect a blood sample and just leave it in a tube for a couple of hours, or if you centrifugate, so if you pull it around in a machine that makes the heavier elements to go down into the tube. Or, for instance, if you compare it to a bottle with water and coins inside, the coins will go to the bottom with the gravity. And the same happens with the blood. So it has the plasma, which is mainly composed by water and some proteins and other elements that are essential as hormones, for example, that are produced in the endocrine system. And then you have this 45%, which is composed by cells, the books, okay? And these cells are essentially, we will see in the next slide which cells are, but in the plasma we have these components that I think you will have access to the slides, so you can later on look into in further detail what is the main function of each one of these, but essentially they have proteins and elements that are essential to the regulation of the body and every cells that are irrigated by the blood stream. And then the cells. And the cells group in three main groups. So the erythrocytes that we call red blood cells. There are cells that are thin with no nucleus, so they don't have genetic information. And they are made to be fast and transport the oxygen, okay, to all the body cells, okay? And so they have a very important protein which is called hemoglobin, which is measured to determine if you have anemia or not. And this hemoglobin is specifically designed to transport oxygen. Okay, so this is the main function of these cells. And then you have the other white blood cells, so this group of cells that are involved in defense, okay, defense of the body with the immune system. Some of them more designed to recognize antigens and have like a first response of innate immunity. Okay, we will see what it is, innate immunity. So it's immunity that it's happening all the time, the first barrier of the immune system. And then some of them produced that work to produce some chemicals that make us to have allergies, for example, and react to some external factors, and others that are specific to like the engineered cells that are targeted to specific elements with adaptive immunity, and those are the lymphocytes. We talked about them yesterday, the B cells and the T cells, those that are engineered, programmed to attack a specific antigen. Okay, that is recognized by the presenting cells. And the presenting cells, the macrophages and the dendritic cells and the B cells themselves can present the cells to lymphocytes, okay? Antigens, sorry. And then you have a third group, which is the platelets. The platelets are fragments of cells, and they are used to produce the clots, okay, and allow us to coagulate. So every time there is a lesion in the blood vessel, these platelets, these fragments just go to the place. There are production of some chemicals that attract them to that place, and they just build a barrier to make a clot, okay? So the heart has 4 chambers, okay, 2 on the right side and 2 on the left side, and this is the typical representation of the blue and the red vessels, okay. The blue vessels in general take the venous blood, and the venous blood that goes to the heart. So the blood that comes to the body to the heart, okay, and this blood enters the right atrium and goes to the left, to the right ventricle, and the right ventricle to the pulmonary artery, okay? So, and this pulmonary artery takes the blood to the lungs to get oxygen, okay? Because this blood has already passed, every time that there is a heartbeat, this blood is entering the heart, and it comes from the tissues, so without oxygen, because oxygen was left there to the cells and the tissues, okay? For them to produce everything they need. And essentially, a vein takes blood to the heart, and the artery takes blood from the heart. Okay? That's how they are called. And in this case, this artery takes blue, okay, blue blood, because it's not oxygen. It's an artery that takes blood to the lungs, okay, to get oxygen. And then there are veins with red blood coming from the lungs, oxygenated, that go to the left atrium and then to the left ventricle. And this left ventricle that you can see here, it's with a high component of muscle because it has to make a big pressure to generate enough pressure to have velocity to the blood to reach every cell in our body. Okay. And then saying this, the heart is connected with two circulation systems, so the pulmonary circulation and the systemic circulation. The pulmonary circulation is the blood that comes essentially from the right ventricle and gets oxygen in the lungs. So it turns from blue to red because it has oxygen. And then the systemic circulation that comes from the left ventricle and goes to all the body and takes the nutrients and oxygens to every cell in our body, okay? And if you get like a zoom in from a capillary, so the thinner vessel in our body, you can see here cells. So cells are everywhere. So the wall of this capillary, this small vessel, it's a group of cells connected to each other. And then inside there is a tube where the blood elements run through with water, okay, which is the plasma, okay? So lymphatic system is also like a road map as well, but it doesn't have highways, okay? So it runs slow. It has also the direction from all the cells in the body to the heart, okay? And it goes to the heart, and I will show you in the next slide, to a connection with a big vein in the top of our thoracic portion of the body. So what it is lymph? This is the first question, I might think. So for you to understand, this is like a tissue. Imagine your skin, for example, where you have a group of cells that are producing like a factory elements that you need to survive. And as the factory itself, it needs to eliminate all the products and the residuals that the cells don't need. And they release them to this virtual space around the cells, where are the blood vessels. And these blood vessels have a specific pressure and a specific concentration inside the tubes that allows these elements to change from this virtual space to the inside of the tubes, okay? If the tubes don't have the full capacity to collect all these residuals. So the remaining residuals and the remaining liquids are collected by this lymphatic system and these lymph vessels. So the lymph is the remaining residuals in the cells, around the cells in the tissues that are not collected by the bloodstream, okay? So eventually, this can be compared like the upper hole in a wash basin. So in the mean that it allows us to avoid an overflow of liquids in a given part of the body. For example, if you do a lymph node dissection when you have a breast cancer or a melanoma, cutaneous melanoma or dorsal cutaneous melanoma, for instance, and you take the lymph nodes and the lymphatics from the axilla, you will reduce the ability of your arm to drain the excess of residuals that are not extracted from the blood vessels. So these residuals will be kept in those tissues. That's why you have lymphedema, okay? The edema that is caused by the reduction of lymph reabsorption. Okay, so if you extract these vessels by taking out the lymph nodes and interfering in this draining system, you will close this upper hole in your wash basin, okay? So, and this is the place where the lymph vessels drain to the upper thoracic veins, and it drains essentially the lymph from all the body to the blood veins, okay, that goes to the heart. And in the middle way, you will find these lymph nodes that I also spoke about them when you make the lymph node dissection and you take the lymph nodes when you have cancer. And in these lymph nodes, you have a lot, it's like a big concentration of lymphocytes and lymphatic cells, and they work as a barrier, so a first barrier. So the lymph that comes here that could have some antigens, some bacteria, and some parts of tumoral cells or some tumoral cells themselves, they are blocked in the first place before going to the bloodstream in these lymph nodes. So they work as a first barrier. That's why some tumors will eventually metastasize first for lymph nodes, because they are drained from the lymph node, from the lymph system, and then they are blocked in these lymph nodes like a first barrier to control cancer. Okay, it's not the case in uveal melanoma, but for instance, it's the case in cutaneous melanoma, where they have a lot of lymph node involvement. And there are also other important structures in the lymphatic system, which are the tonsils, that you can see, and then eventually you can take them out when you are children. And they are nothing else than a group of lymphatic cells as well as a first barrier in the mouth. You have the thymus, which is here just in front of the heart, and it is very important to the maturation of T cells, so T lymphocytes. Okay, and we'll show you later how is it working. And ultimately you have the spleen, which works like a filter. It filters the bloodstream in a big concentration of lymphatic cells as well in the spleen that recognize antigens and bacteria and eliminate them in a specific place. And then let's go back to the cells again. So you have this antigen recognition that we spoke yesterday. The first line is innate immunity. Sorry about that, it's very small. But here you have the mechanisms of innate immunity, so mechanical mechanisms, so barriers. The first cells, so neutrophils, macrophages, and chemical mediators that act for all the external stimuli. So it's innate. You were born with it, okay? And then you have adaptive immunity. And adaptive immunity, and remember the thief, okay? So the antigen is the thief. And innate immunity is dependent on these 911 cells. So the one that you call to go there and that are called by witnesses, okay? The presenting cells, the macrophages and the dendritic cells. And these 911 cells are the T helper cells. So T lymphocytes that help, okay? This T helper will be presented by the antigen presenting cells. The witnesses will show them the antigen, okay, the thief. And they will recognize and they will generate the adaptive immunity. So it's an immunity that is adapted to a specific antigen, new antigen, okay? And these T helper cells will generate two different type of adaptive immunity. Immunity which is dependent from cells, and there will come the T cytotoxic T cells, okay? The T cells that will produce substances that will kill all the bacteria, all the tumor cells that are external and strange for the body. And they will generate effector T cells that will kill this stimuli, and memory T cells, so the registry for the thieves. Okay, they will recognize immediately the same thief every time he tries to enter a house. And then you have the other side, which is the B cell, the B lymphocyte. This B lymphocyte will not produce cytotoxic substances, will not produce chemicals, but it will produce antibodies. So as you remember that slide that I showed you, the antigen and the antibody, which is like a protein that has a common branch and two smaller branches that recognize the antigen and block it. These antibodies will recognize this antigen every time it comes again, okay? That's why, for instance, children have, once they have the first infection, they will get immunity for further contacts with the same antigen. The same with tumors, okay? If you generate antibodies or recognition by T cells for a specific tumor, the immune system will be able to recognize those cells later on. And these T and B cells, these lymphocytes, are the policemen, okay? And so, as I told you before, the thymus is the place where the T cells, the pre-T cells that are produced in the bone marrow, so the inner part of some of our bones that produce the blood elements, these T cells will be maturated and become proficient in the thymus, okay? And B cells are produced and maturated in the bone marrow, okay? And they will be essentially present in lymph nodes and other organs of the lymphatic system, okay? And as you know, the liver is the, so now going out of the lymphatic system, you know the liver is very important in uveal melanoma because it's the main place where metastases go. And there was yesterday a very interesting question about how do we know if the tumor cell is going to the liver or to the lung, and what determines that? Well, essentially, we don't know the answer for this. This is a matter of research for many groups. But there are sometimes some anatomical explanations for it. For example, the tumors that arise from the gut, from the bowel, they are anatomically driven to the liver because the drainage of the bowel goes to the liver. So sometimes there is an anatomical reason for it. For instance, the breast cancer or the melanoma, the dorsal melanoma that I told you before, that drain to the lymph nodes. Okay, they go to the axillary lymph nodes, or the lymph nodes that are closer, they're nearby the tumor. So sometimes there are anatomical reasons, but sometimes, as in uveal melanoma, we cannot explain why uveal melanoma is going specifically to the liver, okay? And it might have some relationship with specific modifications that cancer cell induce and specific targets that the cancer cells find in specific tissues and organs, okay? And what is important about liver anatomy and physiology? So it is divided essentially in two big lobes, left and right. It has these two small lobes, the quadrate and the caudate lobe in behind the back part of the liver. And the liver is located essentially in the right upper part of our abdomen. It can extend to the lower part and to the left upper part of it. But what it is more important to tell you today in this talk is that it have three different drainage systems, or three different tubes inside this organ. So one of the tube systems is this red one, okay? And this red one are arteries, okay? They take blood with oxygen. And this blood with oxygen comes, as you remember, from the left ventricle of the heart, and it is pumped through the aorta, which is the main artery, that is the main highway, central highway that we have in our body, and it goes through hepatic artery to the liver. And it takes essentially oxygen to the liver, okay, to the cells that are in this tissue, in this organ, okay? And other system is the hepatic portal vein system. So it's a big vein that comes from our gut, okay, so our bowel, that absorb the nutrients and go from these veins to the big vein that go to the liver, and where the nutrients that we absorb from our food that we intake are processed by the liver. So this is one important function of the liver, is to process the nutrients that we have with the food. And finally, we have this green system. This green system is the bile ducts. And the bile ducts, where is bile? It's like, again, the sewer of the liver. So the residuals are eliminated in this system, and this system is eliminated to the bowel, to the inner part of the bowel, through bile ducts, okay? And there is a very important circle here between these two blood and green systems that is the enterohepatic circulation. So entero from gut, from bowel, and hepatic from liver. And this keeps sometimes drugs that we give to patients circulating between the liver and the gut, being reabsorbed again, going to the liver, and this is why they are kept sometimes in the body for a while. Another important thing is that is bilirubin. You might have heard about it. It is measured in the blood test. Bilirubin is produced and altered somehow in the liver, and it is eliminated through these bile ducts. And every time the liver loses its function, or the drainage for the bile system is blocked, this bilirubin will be accumulated in the liver, and so will be drained through the hepatic veins and will enter the circulatory system. That's why people who have liver impairment will accumulate bilirubin in the circulatory system, and will get the yellow eyes because the bilirubin and the pigments will accumulate, okay, and will have a dark urine and will have clear stool, okay, because the stool is not eliminating the bilirubin that usually goes to the bowel and is eliminated, okay? So, and as I mentioned, liver is the main place of metastasis in uveal melanoma, and we talked a little bit how cancer originates yesterday, okay? When you have a mutation in your DNA, and this mutation can arise, can generate an oncogene, okay, that has a gain of function, as an accelerates the car against the wall. Remember that image? And can either mutate the tumor suppressor gene, so it loses, so the cell division loses the brakes, and the car is again going against the wall, generating cancer, or it loses the capacity of repairing a mutation. So genes that produce proteins that are specifically targeted to repair DNA damage. When this occurs, you have a first mutation, then a second mutation, a third mutation, and a fourth mutation eventually, and at this point, the tumor growth is completely uncontrolled, okay? And it generates a tumor. And why this tumor is sometimes benign or malignant? This is a very important thing. So sometimes you have benign tumors, so it is an uncontrolled cell division of a specific tissue and part of your body. The difference between a benign and a malignant tumor is that a malignant tumor has the ability to invade other structures, invade adjacent structures. So it's like, imagine your skin for skin melanoma. I think it might be easier to understand. Your skin has an inner part, like a barrier between the out part of the skin and the rest of the body. Every time the tumor grows and overcome that barrier, it invades adjacent structures. So it gets the capability of invade other structures. And for instance, you have basaliomas. They are very common and are not most of the time invasive because they are kept in the skin. As opposed, melanoma is frequently invading other structures. And these malignant tumors, and we do not fully understand how this happens, okay? How the tumor is programmed to invade other structures, and why this invades and the other does not. And the same with metastasization. We know that some tumors have the ability to produce new vessels. So if they build new vessels to the tumor, they are able to put some of these tumor cells into circulation and metastasize to other places. But this is how we see it nowadays, and there are many research to understand why tumors metastasize and why they metastasize to liver or lung, as I mentioned before. But this is how we see it, okay? So, the last part, do regular blood tests detect cancer? I think this is the main question for most of you. At least patients always ask me when they come for an appointment, how is my tumor in your blood, in the blood sample? Can you see it, or how is it going with the treatments? We actually can't say it. So what we saw that it is in the blood components, it's in one part the cells that are produced by our own body, okay, so there's no tumor cells there, and the components, so that we measure. And these components are mostly used to see how is liver working, so enzymes produced by liver, bilirubin, okay, and some other components that tell us how is kidney working, for example, okay? So what blood tests tell us is essentially how our own body is working. So, but if we know that the liver of the patient is affected by tumor cells, so metastasis, and if the blood tests that tell us how is liver going are getting worse, we now extrapolate and then we see, okay, the tumor might be growing. So we should have a CT scan or a PET CT. So this is why blood tests help us in routine practice, okay? And what about tumor markers? Many people ask, how is my tumor marker? As you know, in uveal melanoma, we don't have a specific tumor marker. We do have this LDH, which is an enzyme from the inner part of the cells, which can somehow tell us the amount of tumor burden. So if there is big tumor burden or if there is a big tumoral activity, this might be correlated with a high LDH. But an LDH is not an ideal tumor marker. An ideal tumor marker is a sensitive and specific to a tumor. LDH is everything less specific. So LDH can be increased in inflammatory process. Okay, so that's why we don't have a specific tumor marker for every tumors. Some tumors can be monitored, and response tumors can be monitored with tumor markers, and some of them can be diagnosed with tumor markers, but it's not the case for melanoma. And finally, and I think I don't know if I still have time, 2 minutes. Liquid biopsies. What I have to say you about liquid biopsies is, in first instance, this is not used in routine practice, okay? And it's not used in routine practice because I think we still didn't fully understand how it can be useful for us. We know what are the potential benefits and how we can use it, and we are doing it in many research studies, and it is a very important component in every clinical trials. If we can, we should use it to better understand how we can use it, but it's not essentially used in clinical practice because we still don't know how we can use it. And how it works is a liquid biopsy can be virtually made in every liquid of our body, but it is mainly referred to a measurement in blood, okay, in a blood sample. And what it measures is components from the tumor. It can measure cells, so circulating tumor cells, as we mentioned, and parts of the cells, as we saw yesterday and in this first slide. So DNA, proteins, RNA, okay, that are specific from the tumor, and we did recognize in previous studies that are from that tumor. Okay, so if you recognize it in circulation, we know that there is a tumor somewhere else. Okay, but as I said just now, we don't know where is the tumor. So if we see, like for example, a metastasis in the lung or in the liver, and we don't know where is the primary tumor that originated that metastasis, a liquid biopsy will help us to diagnose it if we didn't see it before, eventually. And but we cannot offer for everybody that comes to a medical appointment and has no cancer or has any clue of a cancer to do a liquid biopsy just to check if there's cancer, because it would be impossible to do. But it doesn't give you a picture of how is the tumor, how the cells that we eventually measure in the cells organize in a tissue. So, and this is given us by a biopsy, a traditional biopsy. And when you take a sample of the tumor, you don't see only the cells that you can measure, or the DNA, or the RNA, or the protein. You see how they organize in a tissue, which proteins are expressed in the cell membrane of the cell, okay, that make us determine eventually where the tumor comes from, okay? If you take a biopsy from the lung and you don't know where is the primary tumor that metastasized to the lung, where are several lesions that you can see in a CT scan, you can determine by a biopsy eventually what is the most probable origin of that tumor with a traditional biopsy. So liquid biopsy cannot necessarily mean that the patient developed metastatic disease. It's also a very important thing. It might also help us know, for example, if you are diagnosed with a uveal melanoma and you do a liquid biopsy, it can be eventually say you that you might have some circulating tumor cells and might benefit, for example, in the future from an adjuvant treatment after surgery or after a radiotherapy. But it does not necessarily mean that you have metastatic disease. It can be correlated with the probability of having it, okay? But we don't know. Moreover, as I mentioned, traditional biopsy is still the standard for the diagnosis of a tumor, okay? And these are some questions that eventually liquid biopsies will help us to answer in the future. I think it's promising, but it's still a little bit far from our clinical practice, okay? So thank you.
Try again. Thank you for giving us that talk. It was really interesting, and I think broke down a lot of different sections of some of the questions that we all have.
I hope that's less boring from yesterday.
Not boring at all.
I was a little bit sleepy yesterday than today.
We're going to have just a few questions, and then if it's okay, if we can send you the rest after this conference today. So if you do have any questions that you've thought about, if you could put them, for those of you that weren't here yesterday, we're moving into the 21st century with our questions. So you can go into Slido, S-L-I-D-O, and type in, it will come up with a hashtag sign, and you type in Ocumel, all in lowercase, and then you can post your questions through there. Put your name on them or not, as you wish. And then we can take those pings. If you think, actually, you can see another question that's hanging, and you think, yep, that's the same as my question, you can like that question, and it puts it to the top, so we know just how popular that question is.
So first question, should we all be having regular liver function tests? So if you go to a GP, okay, so your first doctor, he usually asks liver function tests by routine. It's not defined how frequent it is, but it is part of regular blood tests that you do once in a time. So it's normal to have these regular liver tests, okay? It's just measuring enzymes that are produced in liver and bilirubin, for instance, okay? I was told that ocular melanoma doesn't travel through the lymphatic system. How does it travel? Through blood system, so circulatory system, okay? So there are two roadmaps, so the lymphatic system and the blood system, okay? Is the EST-100 blood marker relevant to ocular melanoma? Well, I don't know the answer for this question. I think we need more experiments to understand this, if this could be a good blood marker, okay? That's all.
And I think that shows that you've answered lots of questions.
Okay, so I just want to say, I don't know if you a few minutes. It's this concept. It's basics. I don't know. I know if sometimes it's difficult and abstract to understand, but they are the basis of our systemic treatments. As medical oncologists, we also think about these processes when we prescribe treatments for patients given IV, for example. So chemotherapy most of the times damage DNA or interfere in the mechanisms of reparation of DNA, generating death of the cells that we want to kill with chemotherapy. But this is why also it has all the other adverse events, because it affects all cells that are in division in the body. So that's why you have hair loss, why you have mucositis and diarrhea. So it is because it's not specific. And the same with immunotherapy. Immunotherapy, it's helping our immune system recognizing these antigens through all this process of antigen recognition that we mentioned, inhibiting inhibitory effects that the tumor have in hiding from the immune system. But this will eventually also make the immune system to recognize and strange our own body system cells. So that's why we have autoimmune adverse events from immunotherapy, because sometimes it is unleashed as well for the recognition of the cells. So that's why we always think in these basic concepts when we are giving treatments for patients. So assuming no surveillance scans, if liver function blood tests are of concern, is it already too late? Well, I'm not sure if I fully understood, but I think that the question is, so if the alteration is measured in the blood tests, in the liver function of the blood tests, is it too late? Is that the question?
I think so, yeah.
Well, it depends on the degree of the altered liver tests. If you see that the liver tests are extremely alterated with high levels, probably it will be difficult to do some systemic treatments, because some systemic treatments have to be adjusted for liver function, as for example chemotherapy, because chemotherapy is processed as well by the liver, and if the liver is not sane, it will not be able to degrade the chemotherapy or process the chemotherapy.
And also,
so it might be late.
I know, I'm thinking if the liver function test is high, if you have an abnormal reading, you can't safely assume that, yes, your liver is invaded with cancer and that's already too late. It just means that there's further investigation needed.
Yes, but the thing is, a surveillance scan, it's difficult to implement because it's more expensive than a blood test. I think regulatory authorities might think about that as a big problem. The thing is that it's not proven as well that a routine scan will reduce the probability of dying from cancer. There's no, generally speaking, there's no such a study that demonstrated that the routine test to detect metastasization or to detect cancer, generally speaking. Of course, there are some specific screenings that are indicated to detect early cancer, like breast cancer or PSA for prostate cancer that can help. But there are no specific scan that has showed that early detection can affect survival. So that's the big question. How, and we need to understand it better and do research to understand how we can protocolate these scans.
Well, if I give you our take on how surveillance is viewed in the UK, if you can then correct me if I'm incorrect in any of those aspects, would that be okay?
Yeah.
So Diogo's mentioned that there's no known evidence to show that actually if you detect the cancer early, that you will then actually live longer. Okay? Now, we would think as non-medics that actually catching that cancer early gives people a better chance.
Yeah.
Does everyone agree?
Yeah.
But there is no data to support that. But that doesn't mean that doesn't happen. It doesn't mean that actually detecting it early won't give you longer. It's just that officially, on paper, there is no data to support that view. So it's just a bit of a common sense approach that actually if you detect it earlier and then you're able to have treatment, that should help you to live longer. So when a doctor says there's no data to support that, it doesn't mean that that's not true.
Yeah. For instance, we talked about the importance that ophthalmologists to recognize uveal melanoma in an early stage.
Yes.
And sometimes it's misdiagnosed by other incorrect interpretation.
Yeah.
And the same for a CT scan. It cannot detect in detail a metastatic lesion that can be detected, for instance, by an MRI. Okay. So, I mean, it is important to do early diagnosis, of course.
Yeah.
We still don't know for every tumor and for every metastasis how we can scan it as a screening for early detection. Okay. Yeah. So that's why it is important to do epidemiology studies and to demonstrate, as we did with breast cancer, that screening is routinely indicated to early detection.
Yeah.
Of course, we save many people with doing mammographies in an early detection process. So it is important, of course, to implement screenings and to determine which tests are important to early detection. Okay.
And I think that's just it. Is that certain things that we would view as normal as a non-medic. Mm-hmm. When we hear a medical person say there's no data to support that, we sometimes can assume that means we were wrong in our thinking, but actually it just means that from a doctor's point of view, there is no evidence behind that.
And just to corroborate that, when we see a lesion for the first time, we don't know for how long it has been growing. Yeah. So even if it is a small lesion, and if you caught it earlier, it might be a UM with bad mutations that will grow really fast. And on the other hand, we can have a lesion that has good prognosis mutation, but has been there for years, and hence it has the same size. So it has a big role of detecting it earlier than than later.
For example, with breast cancer, that I practice a lot, and which is easier because it's more frequent and you can easily do studies, okay?
Yeah.
There's no evidence that shows that you should do an ultrasound of the liver or an X-ray or a blood sample. It's not recommended at all by meta-analysis guidelines that you should do blood sample every 6 months or every 3 months, and people struggle a lot with that. So are you just seeing me? Are you just seeing my breast and hearing my lungs, and you don't do any blood tests? Because it's not proven that that early diagnosis of a recurrence will affect the survival.
Yeah.
That's difficult sometimes to explain, but it's the way you have to manage as well the surveillance.
Actually, Neil Pearce will be joining us soon, who's our chairperson for those of you that haven't met him, and I think he'll probably touch on this a lot more, that actually if something is detected, what can that mean?
Yeah.
And what are the chances that it is something serious and something else that's just quite random? So yeah, thank you.
Hi, I mean, I think you're talking generally across various cancers, but we've seen studies that demonstrate, and this is, in controlled studies, that liver resection, which is often, hopefully, something that people are able to obtain if they have metastases to the liver, has an overall survival benefit if it is detected earlier. And they've looked at people who were detected. So there are isolated studies within different cancers that show that early detection is better, but perhaps generally not overall. So we've kind of focused on those where we've got evidence that we can demonstrate.
Yeah, that's an excellent point. So what should make us think is that we should design a trial that compared a regular surveillance with an ultrasound surveillance or an MRI surveillance, for example, of the liver. And then we will have data to argue with authorities that it is important to fund an MRI. If it is proven, then an MRI will provide early detection as compared to ultrasound or to blood tests. I think this is why it is important that we all work together to join forces to see where is the problem and design evidence to show to those who decide where the money goes to have free access to the recommended screening exams. Otherwise, it will be just doing it randomly, as we think it might be the best for the patient. I don't know if I did explain. Yeah. Okay. Yeah.